Hi, my name is Karen Morley. I'm an application scientist with BioLegend. I've been at BioLegend for over 10 years and I help customers find solutions to their technical and sales related concerns. At BioLegend, we offer a large selection of reagents for flow cytometry. So I wanted to share some tips to help flow cytometrists get the most out of their flow cytometry experiments. And today I will be talking to you about biological controls for flow cytometry. Prior to coming to BioLegend, I worked at an academic flow cytometry core where I was responsible for training new users on how to use the cytometers as well as basic flow theory. So in that role, I would often stress to new users that flow cytometry is a relative technique. You have your unknown samples, and in order to get any information about those samples, you have to compare it to a reference. And the onus is on the person running the experiment to establish the appropriate reference for your unknown samples. And establishing that appropriate control is the difference between sound data and not so sound data. So in a separate video, my colleague Teresa covers instrument controls, which allow the user to reliably distinguish positive signal from negative signal on the instrument. But once you're able to do that, you also want to be able to reliably tell whether or not the positive signal that you're seeing for your sample of interest, your unknown samples, whether that signal is truly positive or whether the negative that you're seeing is truly negative. And this is where biological controls come in. Biological controls can be classified in three ways. You have your negative, positive, or longitudinal controls. Biological negative controls are controls that are non-expressors of a marker of interest. They're known to not express this marker. So they would be used to establish what no signal looks like for your sample type or your experimental conditions. Some examples of negative controls include cells that have had the expression of a marker knocked down by RNAi or CRISPR technology. They can be cells that are known to have lower expression or no expression of a target. You can also use unstimulated or irrelevantly stimulated cells in the event that you're doing a stimulation or functional assay. Or if you were doing a drug study, you could use cells that weren't treated with drug as your biological negative control. For biological positive controls, so these are controls that are known expressors of a marker of interest, and these are used to establish what positive signals look like for your sample type or for your experimental conditions. And examples of positive controls include cells that have been transfected to overexpress a marker. It could be cell lines that are known high expressors for a target that you're interested in, or it could be a sample that's been stimulated if you're doing functional assays, or if you're doing drug studies, it'd be a sample that you've treated with drug. And finally, for your longitudinal controls, so these are controls that are used as a quality measure if you're doing long-term studies where you're looking at the expression of markers over time, like over months or years, you would use a longitudinal or a reference control. And this, good example of this is our Vera cells. These are lyophilized cells that you can use to do this. So we have seven different types of Vera cells, from the basic PBMCs, your control PBMCs from a control population, or leukocytes. There's also PBMCs that have been activated that you can use in stimulation assays, or CD34 positive PBMCs, which can be used if you're doing myeloma or leukemia studies. At the end of the day, your flow cytometry data is only as good as your controls. So talk to your flow core manager, talk to your technical application scientist from BioLegend to make sure that you're including all the necessary controls to optimize and get the most out of your flow cytometry experiment.